Join as we start. I'm Dr. Manasi Golkar, Assistant Professor, to welcome you all at this national webinar on computer-aided drug design and overview organized by Oriental College of Pharmacy. As a brief introduction to this webinar, I would like to say that we have particularly chosen this topic as there is an unnecessary fear about the word CAD, a popular short form for computer-aided drug designing, in the minds of students as well as some teachers. It is looked upon as something very complex or as a part of the domain of computer experts. Hence, I think with this idea in mind, even the Pharmacy Council of India is taking steps towards educating and popularizing this subject by including it as a compulsory topic in the syllabus of final year B.Pharm. CAD definitely helps in prospective research than retrospective research. What I mean to say is it helps in predicting certain properties before synthesizing a compound rather than testing the compound after its synthesis. It has definitely shortened the time period for drug discovery. Now, I am no expert in this subject, so I leave it to our speaker for today to enlighten us with the basics of CAD and its advantages to us. At the beginning of this seminar, I hope all of you must have had a video tour of our institute. Now, I would like to introduce our principal, Dr. Sudha Rathod Madam, who has been a constant source of encouragement behind the functioning of this institute. Mrs. Sudha Rathod completed her B.Pharm from Indore University, M.Pharm from Dr. Hari Singh Gaur University, Sagar, and PhD from C.U. Shah College of Pharmacy, SNDT Women's University, Mumbai. She has worked on target-oriented drug delivery system using microspheres, liposomes, and erythrocytes as carriers for drug targeting during her doctoral studies. Madam is a recipient of Junior Research Fellowship by UGC during her M.Pharm and Senior Research Fellowship by UGC during PhD tenure. Madam also holds a diploma in yoga from Dr. H.S. Gaur University, Sagar, Madhya Pradesh. She has been regularly practicing yoga for more than 20 years now. She has more than 30 publications in national as well as international research journals and 2 patents to her credit. Sudha Madam has also presented her research work at the Control Release Society Symposium held at Washington, D.C. in 1993. Her areas of research include liposomes, nanoparticles, microspheres, sustained release tablets, in situ gels, transdermal films, control release tablets, microemulsion based drug delivery systems, and film coated tablets. She has worked on many projects sponsored by UGC and University of Mumbai. Madam is an approved principal of University of Mumbai and holds this post since last 20 years. She is a member of various committees of University of Mumbai, which include Results Moderation Committee. She has been appointed as Vice Chancellor nominee in governing bodies of many colleges affiliated to university and is also the convener of the Local Inquiry Committee of University of Mumbai. Apart from her academic experience, she has also worked in analytical and production department in pharmaceutical industries for 5 years and is approved by FDA as an analytical chemist. One more facet to her personality is her immense inclination towards spirituality. She has translated 2 spiritual grunts or books from Marathi to Hindi, which are published by renowned publishers. With this short introduction, I now request Dr. Sudha Rathod Madam to say a few words on this occasion.
Thank you, Madam Mansi Madam. I welcome Dr. Prashant and all other delegates, my dear colleagues, for this webinar. This webinar is really very interesting as CAD has become the part of our studies and research. And because due to pandemic, it could be easily possible for us to get Dr. Prashant on online lecture. Though the pandemic has got so many other disadvantages, but I can see some positive things during this period, and it is webinars, series of webinars, which we could manage to arrange in our college. This is one thing. And the second thing is, as far as the administrative part, lot of other things were involved, which include registration, lunch, so many other things, certification. So these all tedious things have been reduced, and we are getting more than more participants than before because of this online webinars. So remaining the quality of lectures same, and all examples, live examples and case studies through this very known academicians and particularly people from industries. We are getting benefited on such advanced topics. With this, I will just speak few words on our college. Our college is founded by Professor Javed Khan in 2004. This college is reaching excellence with support from the management and the dedication of its staff. Our president, who is not there, died in 2020 October last year due to pandemic. But he has done a lot for this college and its development. And now the things are being taken care by our beloved General Secretary, Wasim sir. The college is approved by AICTE and has also received 5 years approval. Since last 5 years, we are getting approval from PCI. Also, this college has got 5 years approval in animal studies, animal laboratories. We have a well-equipped instrumentation facility and also an animal house facilities. It gives me pleasure to share this achievement that this year, Pharm in all their branches have got 100% results in the university exam. And recently, 2 of our students from pharmacology section has got topper position in the university. Our teaching staff at OCP comprises of total 13 doctorate and 1 postdoctorate faculty out of our 21 faculties. Almost all the faculties are university approved, and the college maintains the required cadre ratio as per AICTE. That is professor, associate professor and assistant professor. The college is now a center for doctoral studies in pharmaceutics and guides master students in 3 branches of pharmacy, which are pharmaceutics 15 seats, quality assurance 15 seats, and pharmacology 10 seats. Currently, the college is involved in university projects and industrial consultancy projects. I also have registered 2 patents till now, and 3rd is in pipeline. We have also received funding of rupees 12.5 lakh last year through Lubrizol Private Limited under CSR category. We have on our credit, our faculty have published 28 national and international publications in this academic year. Since last 4 to 5 years, our students are securing university topper position in different streams of Pharm and Pharm level. We have a very strong placement cell. This year, we have organized a 9-day skill development program, which was funded by Rubicon Research Private Limited. I'm happy to share that 58% of our students are being selected by companies in the academic year 2021, and more than 80% of our students of Pharm who have just passed out as far as semester 1 and 2 is concerned, and their thesis is yet to be submitted, but got the job. 80% of our Pharm students got the job. In the current year, we have conducted series of webinars and workshops related to technical knowledge and soft skill development. We also conduct self-defense workshops for our girl students. The senior and experienced faculty at the college conducts free of cost GPET classes for our students. Our students are all-rounder, and apart from the excellence shown by our students in academics, they do indulge in extracurricular activities and excel even on that front. So this short speech, I will finish my words, and we'll start with the webinar. Over to Nutan Madam.
Thank you, madam, for your speech, your kind words. I now request Dr. Mrs. Nutan Rao, head of the Department of Pharmaceutical Chemistry, to introduce our speaker for today. Before that, a kind request to all the enthusiastic participants to post their queries or questions in the chat box. We will have a question answer session at the end of the talk. This is in the interest of all the attendees so as to not break the continuity of the talk. Another request is to keep yourself muted during the entire duration of the talk. Over to you, Nutan, madam.
So good afternoon, one and all. And Mansi Madam, thank you. To know that we know what we know, and to know that we do not know what we do not know. That is true knowledge. When it comes to computer-aided drug design, we need thorough knowledge, and we are blessed our resource person, Dr. Prashant. I'm glad to introduce him to the audience. Dr. Prashant Harper is currently professor in Department of Pharmaceutical Sciences and Technology, Institute of Chemical Technology. He has completed his Pharm from NDMBP's College of Pharmacy and Pharm and PhD in Pharmaceutical Chemistry from ICT. After PhD, Sir has obtained postdoctoral experience from Bellstate University, Detroit, MI, USA. He was first All India rank holder in GATE 1998. He has got several years of industrial as well as academic experience, including the experience at NIMS College as associate professor. He was involved in many consultancy projects as well as research projects. Dr. Prashant has more than 70 national and international publications to his credentials. Several awards, including the travel grants, have been bestowed upon him. His research areas include design synthesis, biological evaluation, and preclinical development of new chemical entities as therapeutic agents, study of biofilm forming and codon sensing inhibitors in gram-negative bacterial, discovery of tubercular agents targeting MTB GUAV2, discovery of novel cancer stem cells inhibitors, computational prediction of toxicity properties, development of novel fluorescent probes for biomedical applications. He has been invited as a speaker on several occasions and has attended many conferences, seminars, and training programs. So with this short introduction, I would like to request our resource person, Dr. Prashant Harper, to begin the technical session. Over to you, sir.
A very good afternoon to one and all present here. I'm very happy to be here in this afternoon. I'm sure you all are awake after probably a heavy lunch, but I'll try and make sure that you still remain awake and enjoy the session. Okay, so as far as so can someone make me the co-host or something so that I can share my presentation?
Yes, Suraj sir.
Okay, thank you so much. I'll just
Yes, sir.
share.
Go ahead.
Okay, perfect. Thank you so much. Is it visible? Is the presentation visible?
Yes, sir. Yes.
Yes. Okay. I'll just put it on the presentation mode. Okay. Thank you so much, madam, for kind introduction. Although the field of computer-aided design is quite vast, but today I'll be only focusing on the computer-aided molecular design or CAMD part. Like madam mentioned in the beginning, about the CAD as a subject, which was introduced in the MPharm syllabus as well as I think final year BPharm syllabus, because it is so very important. And it's I mean, it's not that something. I mean, this science is not new, but it has evolved over all these years, probably last 4 decades or so. And we are happy. Like, I'm happy to be part of this evolution. Back then, when I was in BPharm in Nasik, so we used to see um, on a computer screen. We used to have one teacher, Dr. Professor V.K. Moria, who is currently in Amravati, I guess. So he was into computer-aided molecular design, and he had a software named HyperChem. So he used to run some jobs, and then the molecules used to keep moving or rather dance. And then we used to, I mean, there was a restricted access. So we used to watch it from the outside, and we used to enjoy that. And then also during my second year, one of my seniors, Dr. Tanaji Tareli, who is currently a professor at St. John's University in New York, his paper cutting appeared in Times of India along with Professor V.M. Kulkarni, and that time it was from ICT. Both were at ICT. So this was about the discovery of HIV-1 protease inhibitors with the HIV-1 protease structure, the protein structure shown on the Silicon Graphics workstation. And that was rather the changing moment in my life. I decided that I also want to do something like that, and I wanted to go to ICT. So then I studied very hard. With God's grace, everything went well. I stood first in GATE 1998, and then there was an unrestricted access to ICT. And then I went to ICT, worked with Professor V.M. Kulkarni for masters, and he was due for his retirement. When I completed my master's, it was only 2.5 years. So he was in dilemma whether to take me for PhD or not. So eventually he kind of, I could convince him, and then I became his last PhD student back then, in 2000. Then since he had only 2.5 years before he got retired, or before he was retired, so we had this task of completing it in record time. So I could finish my PhD um, within 3 years, like 3 days to 3 years from the date of my registration. And that's how we could. Of course, he was retired back then. Like 3 months after his retirement, I submitted my thesis, and then next year the viva was conducted. So whatever I learned or whatever I know in this particular field, computer-aided drug design, which they used to call earlier. But sir used to say that you cannot really design a drug. You can design a molecule, you cannot design a drug. Whether that molecule becomes a drug or not, that's a different thing. I mean, there is a very, you all know how many molecules go in preclinical, and then phase 1, phase 2, phase 3, and then how many molecules really come out of the drug discovery program. So that's exactly why I have modified this title. Rather than computer-aided drug design, I fondly call it as computer-aided molecular design, which is CAMD, right? And so as the theme of the today's seminar or webinar is CAMD, an overview. Although I'll be focusing mostly on the medicinal chemistry part of it, but believe me, there is so much to CAMD in general, or computer-aided design. Leave aside molecular part, just say computer-aided design, which is not only useful in pharmaceutical sciences, but people use it all the time in material sciences, in chemical engineering sciences, where people try and use something called as computational fluid dynamics for understanding the manufacturing processes in big reactors. So even before you execute that process in the plant, so that CFD studies can tell you about what should be the baffle design, what should be the dimension, and what should like how the parameters can be optimized, and eventually those studies are very crucial in really making sure that the process optimization happens, right? And then also CFD studies are also very important from the aircraft design. I don't know if you know, when you see, wing of an aircraft, for some aircrafts, part of the tail of the wing is kind of curved. So once I read, that small change in the aircraft wing can save fuel almost like 20% fuel. So you can imagine. So how did they know that? They actually found that out from the computational fluid dynamic studies, and then because when an aircraft flies, so eventually it's in air, right? So air is a fluid. So all those studies, the dimensions of the aircraft and the altitude, and then all other things, the CFD studies actually could tell them about this, right? And in addition to that, very recently I have completely taught the Pharm CAD course. I have taught in my previous institute, but KMK and other colleges, they requested, so I took this complete course. The videos are available in case you would want to know. I mean, if you all want an access to that, I'm very happy to share that. Just drop me an email and I'll share you all the links. Okay. So having said that, now today's topic, computer-aided molecular design. So am I going to make you an expert in CAMD? Absolutely not, right? Because 1.5 hours is a very small time to even talk about the basic concepts. But I'll try and give you an overview as to what these things are and what you should be looking forward to. And now the field of CAMD is slowly now merging with the machine learning and AI, or even deep learning, machine learning, deep learning, and artificial intelligence. And you must have heard or read about many papers or many articles where they claim to have discovered a new molecule with the help of AI. But then some of these are, I mean, exaggeration, but doesn't matter. So we are just going to talk more about all these aspects, and feel free to stop me in case you have a question in between. I mean, let us not wait till the end and then eventually do the question answer session. Please feel free to stop me. Okay. So with this, I'll start with my talk. Now, I usually acknowledge all those who help me in this. So the first thing is, of course, scientific community, because I have taken references and all other material, including my own papers. So obviously the scientific community, and then the organizers of this webinar, Dr. Sudha, Dr. Nutan, Harish, and everyone present here who have made sure that everything runs smoothly. Then, of course, you all participants who have taken time out of your schedule, and you are here to understand, to learn about computer-aided molecular design, right? Although, like I said, I'm going to talk about the medicinal chemistry part of it. I'll again stick to a small molecule chemistry, meaning since most of the drugs which we use are small molecule drugs, I will not really go into the protein therapeutics or biologicals because that itself is a very different field. And of course, the same principles as what we use for the small molecules are also applicable to macromolecules, but it's like altogether a different ball game. This is like a passenger, like commercial aircraft versus a fighter jet. Meaning, if you say that the regular drug discovery aspects of computer-aided molecular design of small molecules is a commercial jet, then the protein therapeutics and the CAD studies, that is a fighter jet. So you can make out the difference, how difficult it is to fly a fighter jet, and just like that. This is just a random analogy to give you an idea. Now, again, like before I start, the views, ideas, and opinions expressed in this webinar are solely mine, right? I mean, all these years, like last 20 years, whatever I have learned so far, whatever I have seen, whatever I have experienced. So I'm going to be very bold in making those definitive statements. But don't mind, as in everyone has their own views and ideas about certain aspects of CAD, but that's okay. As in, this is why we agree to disagree. So don't mind. Okay. So having said that, I would ask you a few questions. Do you wonder it is computer aided and not computer molecular design? Why is that word aided added? right? As in, if it is a computer which is doing everything, then you probably don't need any manual intervention or the intellectual input by humans. But it is not. Fortunately, it is still not the case. Maybe 20, 50 years down the line, it will be the computer which will be doing everything. But again, teaching computer or teaching computer chemistry is as difficult as teaching computer to understand the difference between a digit 8 and 9. So if you go into the if you read more about AI and machine learning, you will understand. So writing an algorithm to tell computer or to teach computer to to identify a digit is far way too far difficult than what we think, right? So human intelligence, you cannot really compare with anything, but of course you may differ from what I'm saying. But then here we are using the computer, we are taking computer's help for doing some really Herculean tasks where you require speed, where you require the graphics and all other aspects, right? So it is a computer aided design and not computer molecular design. So that word aided is very important, because that is going to be the key of what I'm going to talk today. Whatever studies I have cited, my own examples or other examples. So the human intelligence was the starting point, and then the computers further refined the idea or the hypothesis which was put forth, right? So then the next pertinent question is, who designs molecules which may become drugs? Again, this is when it comes to design, and then we are talking about molecules. So the moment you say molecule, you're talking about structure, right? So everything is in the structure. So if you have paracetamol where hydroxy group is para to the acetanilide functionality, but if you move that hydroxy group from para position to either ortho or to meta, this drug, this molecule will no longer be useful as the as antipyretic, or even for that matter anti or analgesic. It doesn't have any anti-inflammatory property, right? So everything is in the structure. So when I say designing a molecule, so that essentially means making changes in the structure or coming up with a structure, a unique combination of functional groups, the substructures, rings, heterocycles, and whatever you name it. And that unique combination is likely to interact with its target, and that is going to give you some activity, because we're talking about, say, pharmaceutical sciences. So in that case, we're talking about some therapeutic benefit or some therapeutic activity, right? So everything is in a structure, and the moment you say molecular design, that means making changes in the structure, coming up with the unique combination of the substructures, functional groups, and this molecule, which is likely to be likely to have drug-like properties. I mean, I'm sure most of you are aware of Lipinski's rule of 5 filter, right? So if you were to design molecules, taking into consideration carbon, hydrogen, nitrogen, oxygen, sulfur, say then halogens, chlorine, fluorine, bromine, iodine, and then sulfur, phosphorus, silicon, boron or beryllium, and then even for that matter, selenium, some of these elements. And if you were to come up with a possible number of molecules, and whose molecular weight is less than 500, or the number of hydrogen bond donors should be less than 5, number of hydrogen bond acceptors less than 10, and log P less than 5, the total number of possible molecules would be 10 raised to 60. Now, and of course, if I tell you that the time elapsed since the universe was created after the Big Bang, I'm sure you are aware of Big Bang theory. So after Big Bang, the time elapsed is 10 raised to 17 seconds. So even if we started right away from the Big Bang, and every second we produced one molecule, we would have still covered only 10 raised to 17 out of 10 raised to 60 possible molecules. So you can imagine how difficult it is to come up with a molecule which is likely to show you all the favorable properties, pharmacokinetic, pharmacodynamic, therapeutic, and not have the side effects, adverse effects, major side effects and adverse effects. I mean, every molecule, every drug has got on an average 70 different side effects. So you can imagine how difficult it is to design a molecule which will actually pass all those tests, all those hurdles, and then eventually come out clean, right? So discovering a drug is not easy, and it's not anybody and everybody's job, and that's exactly why I want to take you to next set of questions. What is drug discovery and development? I'm sure you are very well aware of that. I'm not going to talk more about that, although I have a slide, next slide, which talks more about drug discovery and development, but some of the startling figures. Then next set of terminologies, Chem informatics, bioinformatics, then computer-aided molecular design, which is the theme of today's webinar. And of course, molecular docking. I'm sure most of you, 99% of you would have heard this word molecular docking. And you think that this is very exciting. You take a molecule, throw it in the active site of the protein, and then see how it binds. And then you say that, oh, this molecule is binding to this active site so nicely. So it is likely to act on against that target or on that target. But believe me, this is not so easy. Had it been that easy, everybody would have docked the molecule. Everybody would have come up with the drug. But it is not that easy. And this is exactly what I'm going to talk about, the realities and the, the real life experience of computer-aided molecular design and similar technologies. Okay, so coming to drug discovery and development, the latest figure which talks about how much money it costs to discover a drug is around 4 billion US dollars, which, and if you take the complete Indian pharmaceutical industry, the turnover is around say 35 to 40 billion dollars. So one drug, $4 billion. The whole Indian pharmaceutical industry is $40 billion. Can someone mute the participants?
Yes, yes, sir.
Yeah. Okay, perfect. So it takes around like $4 billion and 10 to 12 odd years to come up with the new drug, out of which 60% expenses go in development. I'm talking about the clinical development, and the rest of them, they go in drug discovery as well as preclinical studies, right? So around 20% on preclinical studies, and then 17 to 20% on drug discovery, the initial part of it. And like the host said that the time and the expenses for drug discovery can essentially be shortened with the help of computer-aided molecular design. That is very true. Okay, so having said that, this I already told you about. The return on investment, like people invest so much money in discovering new drug with the hope that they will come up with a blockbuster drug whose annual sales would be more than $1 billion US, but only few of them actually do that. So the return on investment is awfully low. I mean, if you say only 5%, only 5 of 100 drugs, they are able to bring back the money which has been invested on them. So you can see how bad this, the economics is. But still, pharma industry worldwide makes around $300 billion in profit. It's actually US dollar, $300 billion in profit. So despite these pessimistic numbers or kind of disheartening numbers, pharma industry still makes a lot of money every year. And if you look at the drugs by retail sales every year, so and most of them, they are actually biologic drugs or biologics whose annual sales is in excess of like $20 billion or so, just one drug. Okay, so likewise, small molecules are in the list, but then now that particular space, that if you see top 20 drugs by retail sales every year, a 60 to 80% of those would be biologics, and rest of those would be small molecule drugs. But then there are other reasons for that, and many of these high-selling drugs are patented medicines. The moment the patent expires, they become generic, and their the overall expenditure becomes very less. Okay, so having said that, coming to the molecular design part, like I mentioned, all you need to do is play with the structure. So just like you go to tailor and get the clothes or suit stitched from that. So depending on the requirement, you have to tailor make these so-called new chemical entities, which I'm referring it to small molecule agent. Now these are new chemical entities. That essentially means these are novel molecules. Nobody else in the world has synthesized these molecules. These are not reported in any of the chemical literature. So if you do a SciFinder search, SQL search, you will not find these molecules reported anywhere. And that's exactly why we need, because if I design a molecule, synthesize it, evaluate it, and if it is already known in the open literature, then in that case, I won't be able to patent it. And if I am not able to patent it, then all the money which I'm going to spend on its development is of no use. And that's exactly where this new chemical entity or this novel word is very, very important. So for any project which you deal with, you need to make sure that your compounds are novel. When we were in ICT for our masters and PhDs, so we used to get inside the library, look into the chemical abstracts, and look at the formula indexes and make sure that the molecules were completely novel. But for you, it is just a computer click, and it will SciFinder search or STN search will immediately tell you whether the molecule is already reported or not. That's a different thing, right? So when you are designing a molecule, so what are you trying to do? So you are trying to maximize the potency, meaning a drug which is highly potent is likely to have a dose which is very, very low. Meaning, suppose if you take diazepam or lorazepam, the dose is 0.5 mg, 1 mg, max 2.5 mg. Paracetamol is 500 mg, 650 mg tablets are also available. If you look at sulfasalazine, which is an intestinal antiseptic, its dose is 1000 mg, right? So you can imagine. So if I can develop a molecule which is very potent, so probably I will have less amount, meaning the dose is very low, and because of which I will have to make that molecule in small quantity, and the tablets will also be of. Just hold on. I'm sorry about that. Okay, so potency is what you would be looking forward to. You want to maximize potency. So every time you come up with a molecule, you would want a drug, a molecule which is likely to have nanomolar potency or even less than that. And most of the times we look forward to agents or molecules which bind to their targets non-covalent, right? Because covalent drugs, they have their own issues and toxicity problems. So most of the drugs which we design, small molecule drugs, they bind to their targets non-covalently. But then, if you look on the thermodynamics, from the thermodynamics perspective, you cannot have a molecule which is binding to its target non-covalently and potency less than say picomolar potency. Meaning, you cannot have. I mean, theoretically, it's not possible. So if you want to have, say, femtomolar potency molecule, then it has to be a covalent binder. There are many drugs which bind covalently to their targets. Aspirin is the famous example. Then selectivity. The selectivity is when if there are related targets available or macromolecular targets available, then the drug should bind to only one of them more potently, and then maybe less potently with or with less affinity to the related targets. And this is an issue in kinase inhibitor drug discovery. If you read more about kinase inhibitors, which have actually become so-called molecularly targeted therapeutics, mostly for cancer, as well as some are in inflammation. So you will understand the meaning of selectivity. There are more than around 470 kinases in our body, and some of these are housekeeping kinases. So if you have a molecule which will bind to the ATP binding pocket of the housekeeping kinase, and then there will be some crucial function of the bodily tissues will be inhibited, and because of which there will be more toxicity rather than the useful effect or therapeutic effect. And of course, the next one is pharmacokinetic profile. And for the same reason, you would want a molecule who is clean in terms of pharmacokinetics. Meaning, if your molecule is likely to have a very high first pass metabolism, then that is good for nothing, because the molecule will not be able to reach systematically in sufficient concentration, and because of which either the dose will be high, or even the duration of action will be very, very short. Toxicity issue, like I said, every drug, because the small molecules, they are likely to also bind to off targets. Like I said, 70 side effects for every drug on an average. So you also would want to have molecules which will interact least with the off targets. But again, at times this is not in your hands, and you just have to accept. And that's exactly when in clinical trials, people, I mean, the FDA will look at what benefits, whether the benefits outweigh the risks, and then only you will have the molecule approved for the given indication, right? So then who does all this when it comes to molecular design? So you are actually walking tightrope, and there is a big valley down, and you miss something, and you are meaning that molecule can be will not make it to the next level. Say it does extremely well in preclinical, but then when it goes to clinical, say phase 1 study, and then something is not right and the molecule is dumped. So all this 7-8 years work is gone. Whatever a few billion, maybe few $100 million, which was gone in the drug discovery as well as on the preclinical work, that is all gone. Right. So then who does all this? So it's basically human mind. It's the bright human, or I mean the brightest mind from the human race. They are the ones who, at least at this, I mean when I'm talking this, at least we are the ones who do this. And then we take help of computer for some tasks which require extensive computations, and all the possibilities which are beyond our imagination. And that's exactly where we need a computer. And so we start with the idea, and we take help of computer to make sure to understand of all the possibilities, whether the one which you are expecting is likely to exist. And that's exactly where your hypothesis, you tell computer to test, and the computer will give you the results, and then you interpret those results and come up with whether the hypothesis which you originally started off with, whether it is going to work or not, at least theoretically. Say binding of a small molecule to its target, like that, or it can be combined, meaning there are some tasks which computer will do on its own, right? And nowadays, like I said, the artificial intelligence and machine learning, deep learning has come up. So some of these tasks have been actually automated, but there will be a time, maybe 20 years down the line, majority of the things will be done by the computer itself, even generating the hypothesis. But till then, I will still be talking about computer-aided molecular design, and kind of praising the human minds. But okay, so let us do the next part, that is why of computer-aided molecular design, or in general molecular design, to optimize the drug properties very quickly. That's one, right? Then rationally, intelligently, because you will be faced during this drug discovery cycle, you will be faced and you will find yourself standing at a decision gate. So you have two choices, to go or not go, right? So these computational studies will help you in choosing or taking the right decision, whether if you see that there's some problem with the molecular property profile, predicted or calculated molecular property profile of a molecule. So I would rather not touch that part. I mean, I would rather not make that molecule, and rather than getting into making it and then finding out, oh, it is problematic, I would believe computer. And again, this will come from my own experience, and that's where medicinal chemist intuition will be very, very important. Okay, so just to give you an example, the drug haloperidol, it contains a piperidine ring, right? It's an antipsychotic drug. It contains a piperidine ring. So this piperidine ring is likely to cause a side effect called torsade D points, or Herd channel inhibition, which leads to torsade D points, which is a dangerous cardiac arrhythmia. So as a medicinal chemist, since I know that piperidine containing molecules are likely to have this effect, so I will not have any molecule containing a piperidine ring. Similarly, aromatic nitro group, I will never have any molecule having aromatic nitro group. Aromatic halogens, I will never do that, because those are highly electrophilic and they're likely to cause toxicity. So I will not really touch those, right? So this is what I'm talking about, these informed decisions at various decision gates, and computer aids in taking the right decision. And that's exactly where I mean, we have come, I mean, this field has matured nicely. I mean, our predictions are pretty accurate, meaning the computer predictions and our interpretations of those are pretty accurate, and because of which things move very fast. And that's exactly why we look forward to CAMD, right? So like I said, a use of computer to test hypothesis, mostly generated by bright minds. Like, again, it's not necessarily medicinal chemist. It could be pharmacologist. It could also be a pharmaceutics person, or a formulation scientist who observes some things, and then he or she can also potentially generate an idea. So again, if I talk about pharmaceutics aspect of computer-aided molecular design, you can essentially select optimal combination of the excipient. Say if you're making a solid dispersion, so you have a choice of say 10 different ingredients, and then all you need to do is run some simulation studies, and it can give you an idea as to which of those depending on your drug structure, which of those excipients are likely to do very well when you use them for making solid dispersion. So rather than making say 100 possible combination, you probably make only 10 combinations, and that will tell you, meaning the probability of success will increase by 10 times. Otherwise, you would have made all those 100 combinations and found out like 2 are best, but here you only use 10, out of which 2 are best. So you can see the 10 times increase in the efficiency or the output, right? So when it comes to CAMD, so you are trying to look at the screening of hypothetical molecules. So these are molecules which are yet to be synthesized, meaning your mind has perceived it, right? Many a times we start with a known active molecule, and then using our intuition, chemist intuition, we make changes in the structure. So these are hypothetical molecules because you have not made it yet. You just want to understand if I mean, the possibilities could be like 1,000 or 10,000 molecules, but I don't have time and money to make all of those. So I'll just tell computer, these are 10,000 molecules, which I have perceived, my mind has perceived. Can you tell me which of these are 10 best molecules out of these 10,000 molecules? And this is exactly what the computer will help you in doing that. Many a times we make use of so-called macromolecular structures where molecular docking is used. That is one aspect of computer-aided molecular design, that is structure-based design. If I'm not worrying about the potential target and the interaction of the small molecule with the target, rather I just focus on the molecule structure, ligand structure itself, and then I conduct some studies which come under the purview of ligand-based design. So then I don't have to worry about the macromolecular structure. So those will be ligand-based design strategies. Again, I'll talk about some of these, right? So here, this slide shows you the structure-based drug design aspect, where all you see here is so there is a macromolecular structure, these pipes and these ribbons, right? So these are the secondary structural elements of the macromolecular structure. And then in ball and stick, there is a molecule which is binding to the active site. One, say, if you see on the right-hand side image, the nitrogen of the ligand is interacting with the nitrogen of the histidine 283, which is the amino acid residue in the macromolecular target. Also, the pyrazole ring is interacting with aspartate 351 as well as this glutamic acid 245, and even the pyridine moiety is also interacting with some of. There is this red ball is nothing but the water molecule, which is crystallographically bound or structurally bound water molecule. So if I can go inside the active site of the macromolecular structure and observe how is my molecule binding, and then I also explore if there are any additional pockets in the molecular structure which can be occupied by making suitable changes in the structure so that the potency can be increased. In that case, I would want to do that. And this is exactly what comes under the purview of structure-based drug design, right? In the next case, that is ligand-based design, where we talk about pharmacophore. Now, pharmacophore is the geometrical description of minimum structural requirements for activity, meaning for any molecule to act as a COX-2 inhibitor, it must have X, Y, Z pharmacophoric features located at particular distances. And this is what the geometrical description is all about. And when I have a hypothetical molecule, what I will do is I'll see whether my designed molecule or hypothetical molecule, which could be structurally novel, whether it is matching with the pharmacophoric features. If it is matching with the pharmacophoric features, then I can tell with surety that this molecule is likely to bind to COX-2 and show anti-inflammatory and analgesic effect, right? So this is what. Now here, I haven't really talked about the macromolecular structure, but just by taking making use of known COX-2 inhibitors, I have developed a pharmacophore, and then I generated my own molecules, my own designs, hypothetical molecules. All I have to tell computer is, just check, just the way Amitabh Bachchan tells computer G, isko lock karo, ye karo. So I will tell computer G, you check whether this my set of molecules are matching with the pharmacophoric features. And if that happens, then I'm very happy to actually say that my hypothetical molecules match with the pharmacophoric features, and then that's exactly what I'm going to make and test. And if that works out, nothing like that. And this is where I'm going to talk more about some of my own studies. where you will be able to understand what I did, and how it helped me to fine tune my own hypothesis, and then which we experimentally tested it. Next part is chem informatics, which is a discipline which deals with the chemical information. So anything related to chemicals. So you have a structure, so then you have the shape of the molecule, you have the electronic distribution or electron density distribution around the molecule, then you have the connectivity information, then you have various functional, meaning structural aspects, including the functional groups, and then the bond angles and everything. So this is all chemical information. So that is what is dealt in chem informatics. So here you are representing chemical structure in the form of some numbers, which we call as, say, descriptors. And these numbers can be mathematically used for, say, QSR modeling or any type of mathematical or statistical modeling approaches. Right, so you have a chemical structure and you convert it in some number, and that number can be used essentially to study the structure. And then when you make changes in the structure, how will that affect the numbers and then eventually the activity, right? So this is where chem informatics is all about. And of course, interestingly, you will also be interested in knowing what are the properties of my hypothetical molecule. And this is where I will use chemical data mining approaches to predict the properties. It could be pharmacokinetic properties, it could be molecular properties, it could be physicochemical properties, it could be permeability, it could be solubility, anything and everything. With the help of those models, I can actually predict these properties. So this is where I earlier mentioned the machine and deep learning approaches have kind of been very they are being used by chemists very on a very large scale. If you read the literature, people come up with very interesting models, and the predictions are pretty accurate. And there is a website, there is a collection of these programs at a place called GitHub. So if you are aware of GitHub, you can actually visit GitHub and find out. It's an open source thing, so you will find anything and everything that may be of interest to you. And once you know how to program or how to write codes, you can also contribute. Yeah, but when we are talking about this, as mentioned by the host at the beginning, or the compare at the beginning, that there is a people are afraid, the students are afraid of when it comes to computer aided molecular design. People think that it's all coding. I don't know any coding. Of course, I'm learning machine learning and deep learning. I know some coding, of course, not that I don't know anything, but all these years I have been on the application side of it. I never knew any coding, but I could successfully use all these programs. So this very like what you are afraid of, that you need a coding to understand CAD. Absolutely, that's not a true thing. But if you know coding, you can actually make things pretty easy and customize things as per your requirements. So similarly, bioinformatics deals with the biological information. So where the moment you say biological information, so you are talking about the genes, and then the gene products, and then the whole genome, right? So trying to understand the genetic basis of disease, because that's very important. Once you understand what is going wrong with which set of genes and their protein products, so eventually you will be able to, in a way, deal with that particular information, so that you can use that information for designing better drugs, right? So similarly, differential therapeutic response to drugs by different members of the population, which comes under the purview of pharmacogenomics, meaning same drug, same dose, but the response in different individuals is different. Even the same individual at different times, the response to the same drug and the same dose can be different. So, right, so that's a different thing. Also, you are aware of the isoniazid slow acetylators and fast acetylators, right? So it is the same thing, right? So that's where the biological data mining, like what are those the N-acetyl transferases in some individuals, they are highly expressed. In some individuals, they are expressed to a less extent, and because of which they are slow acetylators, like that. Okay, so that understanding is also very important, because ultimately trying to understand biology is at the base of everything, because we can make any and every molecule, but if you don't understand what is happening at the molecular level on the biology side, we will not be able to comprehend. And this is where computer aided molecular design also comes to our rescue, which I'm going to talk about in some time. So like Chem informatics, machine and deep learning approaches are being sought after for bioinformatics studies as well. And it's again altogether a different science. Unless you are more interested in that, you will not know. As in, you need to read more and more papers. So, like I said, I have witnessed the evolution of CMD at least over last 24 years, which is almost like I would say 60% of what I'm going to talk about. So 1978, the real evolution started where the molecular shape analysis was proposed, and then after that, the first crystal structure, protein data bank came into existence, and then the three-dimensional structures of proteins were available, and eventually that helped in fine-tuning the so-called computer-aided design or molecular design aspects. And the famous examples being HIV-1 reverse transcriptase inhibitors or protease inhibitors. Those drugs, I mean, those who are from my generation would be able to relate to that. 1984, the first AIDS patient was identified, and then it spread like a wildfire. And then there was no medication, just like what we are in this current situation, right? So then there was no medication. So eventually there was this pressing need to discover medication or drugs. And that's where all the HIV, anti-HIV drugs, they made it to the market in like less than 5 years time. So if you read more about all these cases, you will understand how fast it could, people could come up with these drugs, and computer-aided drug design was one of the pioneering technologies back then to have helped the drug discovery researchers to come up with the drugs in record time. So, those are like successful examples, right? So the ligand-based approaches in 1960s, where Professor Corwin Hansch, put forth along with other colleagues or other researchers from Japan, they proposed the concept of QSAR and QSAR modeling, right? That was in 1964. I was fortunate to have met Professor Corwin Hansch in 2005 in Istanbul. I went for EuroQSR. He was 93 years back then. After 3 years, he expired. But then he would still, talk. So 6 days I could interact with him. He was very old, 93 years, but it was a pleasure. Okay, so then slowly, like I said, in the 1980s, the structure-based approaches came to existence with the growing number of protein crystal structures or 3D structures in the protein data bank. Then in the 1990s, the advances in computer hardware, software, as well as computational power further fueled the growth in computer-aided molecular design. Then we had silicon graphics, and then the IRIX and Linux or Unix workstations, which eventually could support the graphics power which was required to visualize and to conduct these studies. I mean, it wasn't possible on a simple computer. I mean, we have seen 486 computers who probably don't know anything less than Pentium 5 or so, but we have seen those days. But nowadays, the graphical power of even our computers is so very high. You don't need some high-end machines to carry out these studies. In fact, the computer which I'm using for the presentation has got 32 GB of RAM, and then it has got all the advanced hardware stuff, including the graphics card, which I use frequently for computer molecular design studies. Okay, so having said that, again, the major impetus to CMD came into early 2000 when the Human Genome Project was completed, and we knew the number of targets which suddenly became available for exploitation or for design. It jumped from 500 odd targets to more than 1500. So that was something which actually was responsible for fueling the growth in majority of these approaches, including artificial intelligence, machine learning, right? So like I mentioned, the technology has matured as we speak, and it is under evolution, but now it is kind of taken over by the AI and machine learning approaches, which is not a bad thing. As long as things work, it is fine. Ultimately, it is computers, right? So it comes under the purview of CMD. So people came up with multiple hybrid approaches. Some people, proposed the structure-based approach combined with the ligand-based approach, meaning pharmacophore-based approach versus docking, and even pharmacophore with excluded volume analysis, where you are making sure that the additional part which you add in the molecule is not bumping into the protein active site, meaning the molecule is able to bind to the target, right? And then there became the sophisticated computational tools, particularly Schrodinger in right from 2005. Schrodinger started with their module on induced fit, and they were the first one to do that. Meaning, when you talk about, say, macromolecular targets and enzyme, those are not static structures. They continuously move, and you are all aware of lock and key theory of receptors or ligand receptor interaction. So when a ligand binds to a receptor, the receptor also undergoes a change in the structure to accommodate the incoming ligand, and that's exactly where the induced fit model became a hit overnight, because none of the earlier tools offered that. Nowadays, it's very commonplace. People have come up with additional tools, right? And then high throughput X-ray crystallography and recently introduced cryo-electron microscopy has made things very, very easy. Meaning, you all have studied TCA cycle, right? And then there are many complexes involved in TCA cycle at various stages. But because those are macromolecular complexes or multi-protein domains structures, it was very difficult to crystallize them and to come up with their three-dimensional structures. But with the help of cryo-electron microscopy, people are now discovering the three-dimensional structures, and eventually, and in fact, I have very interesting videos talking about cryo-electron microscopy. If time permits, I'll show you that video towards the end. In fact, 3 scientists have got Nobel Prize, I think, in 2017 for chemistry in for discovering cryo-electron microscopy. It's fantastic. I'm sure many of you already know about cryo-electron microscopy. In 2017, I went to a Gordon Research Conference on CAD in New Hampshire. In US. So that time, the last lecture was taken by an Indian fellow from Stanford. And that lecture, he spoke only for 45 minutes, but he is I forgot his name, but he was an expert on cryo-electron microscopy. And he stole the thunder. He stole all the limelight just by talking for 45 minutes. All 4 days of lectures on one side, and his lecture was on the other. It was very, very impressive. And believe me, if you know more about cryo-electron microscopy, or if you follow cryo-electron microscopy, that is something you should look forward to. If you get a chance to work on cryo-electron microscopy, believe me, for next 50 years, cryo-electron microscopy is going to be there. There will be many more Nobel laureates in the same field eventually. Coming to CAMD success stories, HIV-1 protease inhibitors I told you about. In fact, if you talk about the fluoxetines, like norfloxacin, ciprofloxacin, so those were supposed to be the first rationally designed set of drugs, but then those were designed using some raw tools, back then using molecular shape analysis and to some extent docking. All these fluoxetine, they act on DNA gyrase, right, which is required for the unwinding of supercoiled DNA. So these molecules inhibit that, and because of which the DNA cannot unwind, and then the genes are not able to express, and because of which the microorganism dies, right? It's a synthetic antibiotic. But then the actual credit goes to the discovery of imatinib as the first rationally designed, or the molecule design using computational studies or computer-aided molecular design. I think it was discovered by Novartis, I guess. Right. So imatinib, which acts on BCR-ABL protein kinase, it is useful for chronic myelogenous leukemia, and it was the first molecule ever designed by computer-aided drug design, computer-aided molecular design. So there's a very nice paper in Nature Reviews Drug Discovery, how imatinib was discovered. It's amazing. It's phenomenal. You should read that paper, and you would know the potential what the computer-aided molecular design holds. right? So in addition to the actual structure-based as well as the ligand-based thing, like I said, the computers can be used for a prediction of the physicochemical as well as toxicity properties using structure and ligand-based approaches. In fact, there are tools available which can help you in predicting. I'll show you some of these tools towards the end. One of the famous tools is Swiss ADME, which can give you a very nice property prediction profile of the any molecule which you would want to put in. There's another one called Protox2, which will tell you the predict the LD50 of your molecule and whether it is going to be immunotoxic, cardiotoxic, whether it is going to be teratogenic, carcinogenic. Amazing. Now, at the back end of these tools or web-based platforms, there are a lot of models. Some of them use the ligand-based approaches, I say molecular, descriptor based, or even pharmacophore. And then there are even approaches, you make any molecule, or say you isolate a novel natural product from some plant or some natural source. You don't know, it is not reported anywhere. You don't know what it is likely to act on. There are target prediction softwares available, free softwares, or other free web tools. All you have to do is just paste the structure, and that software will give you what is going to be the structure potential target for that particular molecule. And then from that you can eventually design your experimental studies and test it in experimentally test it. We have done that for some phytochemicals. I have not included that in today's paper, today's webinar, but then we have done that. We have done all sorts of things. Believe me, if you follow my papers, we have done, you name it, and we would have probably done it. Except machine learning, I haven't published so far, but like I said, I'm learning on my own. So very soon I'll be able to publish that. Okay. And that's exactly where I said the ML and DL, machine learning and deep learning tools, are slowly spreading their wings, meaning they're getting very commonly used by the chemists. Right. So coming to CAMD and the magic wand, which is nothing but, magic wand is like a magician will have the wand and he will just rotate it, and then things will appear from nowhere. Although it's just an analogy, but I would want to say that CMD can at times, if it is used appropriately, it can definitely help you in achieving things which you can even which you can't even imagine. Okay, so this one is from my own study. I was working for Pharm back in 2011-12 before coming to my previous institute. So here, what we did was we were working on this very interesting target called inosine 5-monophosphate dehydrogenase 2. It's actually an anti-cancer target. It is also involved in inflammation. So we were trying to develop it for inflammatory disorder called ulcerative colitis and Crohn's disease, IBS, inflammatory bowel disease, right? Because that's where some immunology component is involved. Lymphocytes are involved. So what this target does is it is responsible for production of guanine nucleotides, meaning the de novo biosynthesis of guanine nucleotides. And lymphocytes are solely dependent on IMPDH for their requirement of guanine nucleotides. So the moment you cut short this or inhibit this enzyme, the guanine nucleotide supply is cut and the lymphocytes die. And so the immune suppression happens, right? So that's exactly what we were studying. So what we did was in Nycomed, a Swiss company, or rather a Dutch company with headquarters in Switzerland. So what we had an in-house database of 900,000 compounds. So what we did was we did a virtual screening using ligand-based as well as structure-based approaches. And out of 900,000 compounds, we selected 50,000 compounds for the actual experimental testing. So all those 50,000 compounds were shipped from Germany to Hyderabad with GVK Biosciences, and they developed a high throughput assay, and they could actually test all these 50,000 compounds. And we actually came up with at least 17 compounds which belong to different chemical classes. And if you follow IMPDH2, you will understand it's a difficult nut to crack. In fact, two of my PhD students actually worked after I came to academics. Two of my PhD students, they worked on IMPDH2. One worked on IMPDH2, which is for the human cancers. The other worked on IMPDH, but from microbial sources for anti-infective property. So this was the thing from my own study. So I'm one of the authors here, Aarti Prabhu, who was earlier working with the Nycomed. So she was also part of the team back then. Then, so again, like I said, when it comes to CAMD, people have, people think that it is again kind of a magic box, but it is not. And everywhere I mention, if it is used appropriately, it can give you hope. Otherwise, it's just a hype. I mean, I'm saying that out loud, since I mean, even if I have worked in this field for like last 22 or more years, 24 years, right? So you have to use it wisely to actually come up with some meaningful results. In fact, I tell you a very funny story. When I joined Nicholas Piramal after my PhD, I was with Bharati Vidyapit's College of Pharmacy. From there, I went to Nicholas Piramal. So they were very excited that I was doing all this computer-aided molecular design. So I gave them the first set of results. So rather than testing it in vitro assay, they were so excited that they directly took it in animals. It was for some anti-diabetic anti-diabetic activity, PPR gamma modulatory activity. I was telling them that things don't work this way, but then they actually went and did it animal, and it was complete failure, right? So then after that they came to their senses, and we could do some wondrous work after that. Okay, so like I was mentioning, when you're talking about CMD, you're trying to oversimplify things which are very, very complex at the biological level, right? For example, ligand receptor binding. So if you have to do ligand receptor binding, meaning when it happens in the body, so the biological target, which is in the aqueous environment, is surrounded by water molecules. When the ligand, which is complementary in shape and electrostatics, which is also surrounded by water molecules because it's in the cell or in the aqueous environment. So when they have to unite together, both these water molecules layer has to go away. That is called desolvation process, right? Without that, if there is energy, if it is energetically favorable, thermodynamically favorable, then only the ligand receptor complex will form. Otherwise, it will not, no matter what you do, right? It will not happen. I mean, just imagine you are into an area where there are a lot of caves, and you have been given a task to explore the caves all by yourself. But the condition is that you will be, there will be a piece of cloth will be around your eyes so that you cannot see. So how will you sense whether there is a cave, whether to go inside, whether to come out or whatever? You will try and see, use your senses, meaning your hands or maybe smell to some extent, but mostly with the help of hands, since you cannot see. So you will try and see whether there is a place to go in or go in which direction. So the same thing happens when the molecule is in, say, from blood, it approaches the site where the target is. The molecule cannot see the target cannot see the molecule. So they have to sense each other with the so-called drug receptor interaction forces, which include the hydrogen bonding, electrostatic interactions, and then van der Waals interactions, hydrophobic interactions, dipole-dipole interactions, London dispersion forces, and all. So you can imagine how complex it is, but we are trying to do as if it is such a nice thing. You go to a friend's house and you talk to a friend, and then eventually go with that friend for playing. I mean, this is very simple, but if you are in a city, your friend lives in that area, but you don't know the address. So just imagine how difficult it would be if you don't know the address, but you have to find your friend. In other case, you know where your friend stays, you know the address, you directly go and meet that person. So the second case, or rather the first case, where you don't know the address, so this is same thing happens here. The ligand doesn't know where it is likely to bind, and this is where the complexity of simulating what happens at the biological level comes into picture, and then CAMD or molecular docking is an oversimplification of that. So no matter what you see that your molecule is binding very nicely in the active site, but I can guarantee you 80% of the times it will not really act on that target unless you would have studied the solvation pattern and other things. Okay, so having said that, it's very interesting for you to understand the limitations and capabilities of these tools, without which you will not be able to. I mean, otherwise it's just a fancy thing where you generate, extremely good figures and things look very nice on paper, but indeed they are not. In fact, I also have some very interesting videos, but all the videos I'll play in the end, and I'll end my talk with those videos, right? So you need to understand how do drugs interact with their receptors. I have briefly told about the desolvation and the thermodynamic. So thermodynamics will play an important role, because when a drug and ligand receptor complex has to form, then the energy of that complex has to be lower than the sum of individual energies of the receptor and the ligand, without which it will not really form that complex. That delta G, free energy change, must be negative. And then when you want to desolvate the water molecules, say the receptor desolvation as well as ligand desolvation, so there is an energy required for that. So that will add to the positive side of the energy, but you want to go on the negative side. So that's where you must have individual interacting groups in the ligand as well as in the receptor, which will overcome that energy barrier. Then only you will be able to find, or other you will be able to make your ligand bind to the receptor, and that's how the complex will form. Okay, so having said that, the next question is, can we simulate what happens at the biological level in silico? Yes, you do, and that's exactly where the the importance of CMD lies. And again, I'm not here talking only about the medicinal chemistry part of it. I told you about solid dispersion. You have a drug, you select the excipients in the ratio which is allowed by the your formulation knowledge, and then you throw it to the computer, run some simulations, and see whether that is likely to give you a good complex or not. Similarly, beta cyclodextrin, you can simulate how beta cyclodextrin and the inclusion complexation with the drug happens. And that can be confirmed again with the help of NMRC, with the help of a technique called ROSI, or even for that matter, NOSI, which part of the ligand or drug has actually gone inside this beta cyclodextrin cavity or not. So all of these can be simulated in silico. Can we visualize it on computer screen? Because if I am able to visualize that, nothing like that, as in, then I'll be able to take better decision as to what I should be putting in the molecule, so that it becomes more potent, more selective. I mean, if you read about COX-1 and COX-2 inhibitor discovery, so COX-2 inhibitors, they have got additional group, meaning they are butterfly-shaped molecules, which actually makes them difficult to bind to COX-1, which has got a housekeeping function. So that's why COX-2 inhibitors do not have the so-called ulcer-causing properties, whereas COX-1 inhibitors, they are highly, or non-selective COX inhibitors, they are highly ulcerogenic, right? And that's exactly where COX-2 inhibitors came into existence. But then COX-2 inhibitors, they have other issue where they cause severe cardiotoxicity. Again, there is a reason for that, and so that's how, so you try to improve the selectivity, but you invite the toxicity. That's what I was saying. It's not easy. It's not. It's very, very difficult, right? So you need to understand which principles govern the conformational or molecular dynamics of the macromolecular targets and their small molecule ligands. Meaning, these molecules, when inside the aqueous environment and the body temperature, they'll always be moving. They'll not be static. And that's where small molecule will go bind to the receptor, come out, bind, come out, bind. And that's where, the receptor occupancy, some of those who are from pharmacology, they would know the receptor occupancy is very important when it comes to drug action and the drug effect, right? So receptor occupancy theory tells you that how much time the drug is actually in contact with the receptor will decide, along with the receptor numbers, which eventually is responsible for the observed effect. And that is where everything is kind of, revolves. So people have come up with the PET scan or other diagnostic techniques where you can actually visualize the receptors with the help of radioactive ligands and all those fancy stuff, but very, very important, because you're trying to understand what happens at the biological level. But computer tells us, computer makes that job easy, and that's why the more the benefit we observe. So likewise, anything can be simulated on a computer screen, including your pharmaceutic stuff or medicinal chemistry or even toxicity targets and everything. Even you can find out whether your molecule is if you have a molecule, whether it will be able to bind to cytochrome P450 and which site on the molecule is likely to undergo, say, hydroxylation. If it is cytochrome P450 3A4, it is possible with the help of structure-based approaches. Although you may say that cytochromes, they have got a very broad substrate specificity, but it can still be modulated, and that's where the induced fit model comes in picture, where we say that the molecule, the macromolecular target on ligand binding will also undergo changes in the structure, which can be considered for effective design of molecules against a particular target. Interestingly, when you talk about the three-dimensional structures, that is X-ray structures, so those are static structures because you are taking a crystal and then subjecting it to X-ray diffraction. But if you talk about the solution structures where NMR can give you more information, but again, that's a very special science which needs very highly sophisticated NMR instrumentation. And that's where the cryo-electron, recently introduced cryo-electron microscopy is making waves, or rather making substantial progress in uncovering the biological mysteries, meaning a lot of things we don't know about ourselves till date. We don't know how neurons actually develop and how when the fetus is in the womb. That's exactly where our understanding lacks, and we are just trying to understand things which we don't know about, and the computer-aided molecular design in some context can be helpful. So this is again one of my papers where we tried to use molecular docking and designed the new chemical entities. This was published in European Journal of Medicinal Chemistry. So this is Chetan's work, my PhD student who works in industry now. So again, we continued working on human IMPDH2 for cancer. So here we came up with a very interesting set of molecules which were novel. And then we actually, like I said, IMPDH is a difficult nut to crack. So we use molecular docking for optimizing. So you can refer to the paper. I'm not going into the details because in 1.5 hours, it's not possible to cover everything. And that's where, so the first part is the natural ligand as in the known inhibitor of human IMPDH2, which is a mycophenolic acid. It is used as a drug. It's already in the market. It is used for organ transplant rejection. Right. So what we did was taking clues from mycophenolic acid, we developed our own molecules, novel molecules, with the help of molecular docking. So these are like B and C are different molecules, and D is all three together. So we tried to learn from what happens at the in the structure, and eventually put in our own thoughts, came up with the molecules, and we could actually design molecules which were slightly more potent. I'm using the word slightly, it wasn't upside down, which were slightly more potent, or rather, I would say, slightly less would be probably the correct term. They were close, but then we could actually do that, and none of these molecules were reported in the literature. So that's where we use the CAMG for designing with the help of molecular docking for this particular case. In the next topic is comparative protein modeling or homology modeling, which is another very widely used area of computer-aided molecular design. Like I said, after completion of Human Genome Project, more number of protein targets were available for exploitation in a disease process. But then the speed with which the three-dimensional structures are determined, say using X-ray crystallography, is very, very slow. The PDB contains a little less than 200,000 structures. And then there are more than large number of proteins whose structures are not known, and not every protein can be crystallized and whose three-dimensional structure is determined. So in that case, again, we look forward to computer for predicting the three-dimensional structure of the proteins. Right? I mean, it's very, very difficult task, believe me. Just to give you an example, I've given it in earlier webinars as well. If I have to simulate how a protein fold in a body, which happens in millisecond, with the help of fastest supercomputer today, even if I take only a protein of amino acid 100 amino acid long, it will take me more than 200 years to simulate with the help of fastest supercomputer in the world to simulate. I mean, I'm not going to live for 200 years, so I will not know whether the computer really did that job correctly or not. but then that is so again, things which happen in few milliseconds in body, the fastest supercomputer will take more than 200 years. So you can imagine how difficult that task is. But here, what we try to do is, here we try to look for proteins whose three-dimensional structure, the primary sequences similar to a protein whose three-dimensional structure is known. So we are just trying to use the protein whose three-dimensional structure is known as the template for coming up with the three-dimensional structure of the protein whose 3D structure is not known, but whose amino acid sequence is similar to the protein whose 3D structure is known, and that's why it is comparative protein modeling. So this paper we have published in 2019. So this is a target system XA antiporter, which is a target in epileptogenesis or epileptic seizures. It is also a target in glioblastoma. There's only one molecule which is known, and that is sulfasalazine, which is highly acidic drug which cannot penetrate brain. But in vitro studies, they have shown that it is a potent system X inhibitor. So we wanted to discover molecules which are more potent than sulfasalazine and which can penetrate the blood-brain barrier. So, but then the hitch was, because it's a membrane protein, the structure was not known. There was one homology model structure which was available, but it wasn't really that useful. So what we did was we approached this researcher from Australia, Neha Gandhi, who had access to a supercomputer. So she did the comparative modeling part, the comparative protein modeling part. So we built the homology model, and then she subjected it to molecular dynamics with the fastest supercomputer in the world, which she had access to. And it took around, say for 2 nanos, well, it was 600 nanosecond dynamics she ran. It took around, I guess, 4 weeks to complete that job, as in she used to get time slots. So it took around 4 weeks to complete that 500 or 600 nanosecond dynamics. But then finally, we actually came up with this very interesting structure. And then this structure was not known, meaning it is probably the best structure which is known in the literature so far. Not that because we did it, it's just that it tried to address some of the issues which were there with the earlier structure. Only one homology model was reported. Then we used this structure to design molecules which were much better than sulfasalazine. This is A is sulfasalazine, and these are the other ones which are much better. Now you may say that this is an azo linkage and this is a bad one, but that's okay. As in, we just tried to chop off the sulfasalazine and see how it worked, and then we came up with our own versions which were much better than sulfasalazine. You can refer to that paper. So here, what we did was we used comparative protein modeling, which is one of the techniques of computer-aided molecular design for coming up with the three-dimensional structure of a protein which was not known. And then we used that structure for molecular docking and developed and designed and developed our own molecules. Although some of these molecules were good, but a lot work still needs to be done. But that's where the scientific community will take over. And in fact, this paper has been cited quite a lot of times. Because there were only two groups. There was one group, I think in US, some Patel and Patels, and the student who worked on this, my student, I was a co-guide on his thesis, Dhaval Patel, he was also Patel. So only Patels were working on this protein so far, and now there are many groups which have started working because it's one of the prominent targets in glioblastoma multiforme or GBM, which is a dreadful CNS cancer which cannot be cured. So we need drugs which can be cured, which can be helpful in curing GBM. Then comes the next approach, ligand-based design, where you essentially use, you don't worry about the macromolecular structure, or even if you don't know whether the macromolecular structure, that's fine. Only you deal with the known active molecules of the that particular with a particular therapeutic activity, and you use that information, develop some ligand-based approaches, including pharmacophore elucidation. So this is one of my own papers, which we published back in 2008 in Journal of Medicinal Chemistry. This was my second paper in Journal of Medicinal Chemistry. I've got 4 so far. One is review, and the other 3 are Journal of Medicinal Chemistry is the best journal for medicinal chemistry. Okay, so here what we did was we developed molecules for dopamine D2 and D3 receptors for modulating the effect for Parkinson's disease. And then some of these molecules were also used for binding with dopamine transporter that was a different thing. So what we did was we tried to develop the molecules which were kind of mimicking dopamine, which is the neurotransmitter, and the natural ligand of dopamine receptor. So we designed molecules. So for that, we took the active molecules known for D2 and D3 receptors, developed the pharmacophore, and then designed our own molecules, and then came up with very potent molecules which could show dopamine D2, D3 receptor binding. Right, so you can see on the right hand side, these are some of the molecules which we designed and which actually did well. In fact, my postdoc boss, he has taken one of these molecules, not exactly the one shown here, but from the same project, he has taken it further, and it is under preclinical development, or so he has licensed it out, licensed it to somebody. That person is working, that company is working on that, right? So this is where you are not worried about the three-dimensional structure of the macromolecular target. Rather, you only focus on the active molecule. Use that information, because when I say a molecule is active, meaning it is likely to bind to the target. So rather than working on that particular target itself, I take the information which is present in that active molecule structure and use that for designing molecules which will similarly bind to the target and eventually show the activity. I mean, this is the easiest approach, because many a times the target is either not known, or it's not possible for you to come up with the target structure. Right, the other famous ligand-based approaches are QSR-based approaches. This is again my first paper from PhD work in ICT with Journal of Medicinal Chemistry, in Journal of Medicinal Chemistry. So here what we did was we did three-dimensional QSR studies for 1,4-dihydropyridines. So this one, the work was, the experimental work was done by Professor Anamik Shah's group, who was, I think, who is a vice chancellor in Gujarat Technological University or someplace. Right, so his group synthesized this 1,4-dihydropyridines. I did the 3D QSR work. Okay, so here what we did was we used the COMFA and COMSIA, which were the three-dimensional 3D QSR approaches, and then we came up with we identified the features in these molecules which were likely to increase the activity. We tried to tried to understand the basis. The molecules were already synthesized and tested. So in retrospect, we tried to understand why were active molecules active and why were inactive molecules inactive. And this is what, these are the contour maps. I'm not going to explain you all this, but then, so the QSR-based approaches basically use only the ligand information, and depending on whether it is a 2D QSR approach or a 3D QSR approach. So if it is a 3D QSR approach, you're trying to deduce the structure of the hypothetical target to which these molecules likely to bind. We did not know where these molecules actually acted, or where that which target was which was a molecular target for these molecules. All they did was they tested the anti-tubercular activity, and that's how they came up with that. Okay, so again, in the same, we also use the QSR, 2D QSR based approaches, where we calculated molecular descriptors for all the targets and tried to correlate with the activity, meaning the anti-tubercular activity. And then we came up with many good models. And these models actually told us what you should be doing for making changes in the next molecule so that it becomes active. So once I have a set of molecules, I do all these ligand-based, structure-based approaches wherever it is possible, and then use that information for designing molecules which are likely to be more active. Right. So I'm just trying to go in the direction which will give me more and more potent and selective molecules. And this is what the magic of CAMD, or computer-aided molecular design. Okay, so the next important part is prediction of physicochemical properties. Swiss ADME is one of the, in fact, I'm going to show you a demonstration towards the end, if time permits. So here, all you have to do is just give the SMILES notation of the molecule, or draw the structure of the molecule, and it will predict a large number of properties for you. It will predict what is the log P value, like this is all predicted values. Then what is the solubility, what is going to be the class, whether it is poorly soluble, highly soluble, moderately soluble, then GI absorption, blood-brain barrier permeability, whether it is going to be a P-glycoprotein substrate, which is an efflux, PGP is an efflux transporter, then whether this molecule is also going to inhibit the CYP isoforms, which are mostly responsible for metabolizing most of the drugs, CYP3A4, then 2C9, 2C19, 2D6, 1A2, all those things. And then whether the molecule also follows Lipinski's rule of 5, what is going to be the bioavailability score, and then other few things, right? So this is again freely available. You can have predictions for your own molecules before you even synthesize them or take up for synthesis. If now you see this is a radar diagram, all the majority of these points are outside this radar. That means this molecule is not actually good. But believe me, as in this is a drug. The name of this drug is lumefantrine, which is an anti-malarial drug, which is not drug-like, but it is still used, not necessarily that. But these are all predictions, so we can still take them with a pinch of salt. Okay. The other one I was talking about using so-called Protox2, where it can predict the toxicity. So here it is telling you what is the predicted LD50. So pharmacologists, this could be potentially useful. All interesting thing is you have to give a structure of the molecule. It will not tell you what is the LD50 of the extract. No, it will not. You have to give the exact molecular structure. And it says what is the predicted toxicity class. It's 3. The average similarity with the molecules which were in the database is around 50%. Prediction accuracy is around 67%. So it will also tell you to what extent you should believe these results or not. And in fact, they are improving. So all you have to do is just, in fact, I'll show you if time permits Protox2 as well. But then, in addition to that, you can predict the immunological toxicity, the carcinogenicity, teratogenicity, many toxicity endpoints for molecules you can predict, and this is amazing information. Likewise, there are tools for predicting the blood-brain barrier permeability of molecules. There are tools for predicting, I told you already about target fishing or understanding the potential target. So all of these approaches use some model or some structure-based approach, meaning ligand-based approach or structure-based approach for coming up with the prediction. And most of the times, at least it gives you some information which is more likely to be accurate, because these models have been tested and validated. So prediction accuracy is definitely there. Not that it will tell you everything accurately, but at least it will give you some starting point for your further research. And this is what I would say the most interesting part. So it will give you some food for thought, which you can build up on. You can design your experiments based on the predictions, just like we did in one of our papers, which is not included in here. The last part is drug repurposing. Again, with the help of computer-aided molecular design, you might have read about a lot of these studies, but this one is from my own experience. In fact, we have also done the same thing for COVID-19. Okay, I'll tell you about that. Okay, so coming to this study, this paper was published in 2016. So there was an outbreak around 2015 of Ebola, particularly in Europe, and everybody kind of woke up. So we thought of undertaking this computational exercise for discovering anti-Ebola drugs. So what we did was we used the three-dimensional structure of one of the proteins of Ebola, which is called as VP35 protein. So we took the ligand of that VP35 protein present in the crystal structure and did the shape-based and electrostatic-based similarity search. So it was in a way, I would say, a ligand-based study. So what we did was this is the green ball, and the green stick model is the VP35 ligand. So what we did was we did that we took that molecule out of the crystal structure and calculated the shape parameters, because this was a crystal structure binding pose. So we took the shape parameters. Now you see this gray color shape, finger-like shape, and these are the pharmacophoric features. The green, all these ball-like structures are the pharmacophoric features. Then we also tried to look at the electrostatics part. Like I told in the beginning, when a receptor has to bind to the ligand, so they will bind by the, of course, the shape similarity or shape complementarity, and then next part is the electrostatics part, right? So we tried to use this information, the shape-based information and the electrostatics-based information, to come up with molecules which eventually could bind to VP35 of Ebola. And this is the three-dimensional structure of the VP35 protein and some of the molecules which we, so then here, because it was drug repurposing exercise, we used the known drugs from drug bank for potential binding with the shape and the electrostatic complementarity. And the hits, the top hits were eventually docked in the VP35 protein. And this is where the combination of both the structure and ligand-based approaches happen. And so, but then when we communicated this paper, fortunately, we found one paper in which they actually screened almost like 600 drugs for anti-Ebola activity, not exactly VP35, but anti-Ebola activity. So what we did was we tried to compare the list, and we could actually, so there was an overlap. There was an overlap between our list of hits and their experimental hits. And that's how we could convince the editors of RSC Advances as well as the reviewers that our studies actually, even though it was a computational study, but we could still come up with very interesting predictions. And that's how they accepted this paper. Right, because I mean, doing computational study is one, but the experimental validation is another aspect, and that's what every journal would like to ask. And that's exactly the point: is you come up with the hypothesis and then use that information to design the experiments, and then show finally that your computational predictions are more likely true with the help of some experimental evidence, right? So then, although it doesn't, you can't really see here, but what it reads is, did any of this really work? Meaning, I can talk for hours and hours or days, and then finally, you may ask this question, oh, you talked so much about this, but I can consider this as it's a purely or sheer academic exercise where you got papers out of it, but any of this really go into the market or other clinic, not exactly market. Yes, indeed. I mean, we are very proud to say, I'm very proud to say that on 10th of August, that is last month, I got my first US patent. So this is about the anti-cancer compounds. This is my first patent ever, right? So this molecule, we have already filed IND. We got permission from CDSCO for conducting phase 1 clinical trials. Of course, this molecule belongs to Godavari Biorefineries Limited. I have been associated with them as an external consultant for almost 8 years now. This is 9th year going on. So they had a natural product, and with the help of computer-aided molecular design, we could modify the molecule, make it structurally novel, and then file the patent. The US patent is granted. Indian patent will be granted sometimes this year. The last hearing is scheduled on 4th of next month, 4th of October. Then Australian patent, Japanese patent, and the EP patent, they're at various stages. This is just one. So likewise, there are other two more series for which we have filed the PCT application already. And the same molecule, which is part of this patent, we are going to file, I mean, they are going to file the US IND sometime this month or early next month. And then the phase 1 studies will also begin. On the similar lines, during COVID time, we also came up with a molecule who could be potentially antiviral molecule, which they eventually got tested. The hypothesis came from the computational studies. They tested it initially here in India. It showed good activity, very good, as good as remdesivir. Now you may say that remdesivir doesn't act, but then of course the targets are different. But antiviral activity was there. This was also sent to NIH. They also confirmed it, and then after that it was tested for efficacy studies in golden Syrian hamsters. SARS-CoV-2 animal model, and it did show very good activity in, I would say, in the preventive mode. Meaning before the infection actually was introduced or inducted, this molecule was given. So the best protection was obtained in the preventive mode. It did show good activity in the treatment mode, meaning the molecule, you treat the animal with the molecule after the infection was induced. So we have filed the IND for that as well. Hopefully that gets also approved soon. We'll have two molecules in phase 1 trials. The first of this molecule, the one which is part of US patent, so it will start, the studies will start in, phase 1 studies will start in patients of triple negative breast cancer, right, which is a very aggressive cancer in Indian women worldwide, but Indian women mostly, right? 50% of the patients of triple negative breast cancer, they do not respond to any therapy, right? So this is where we would want to, say very proudly that the molecule the phase 1 studies will start. Although this is just one patent you see here, but 8 years of hard work has gone behind that, more so from the earlier team of Godavari Biorefineries Limited, which is a Somaya group of companies. They are located in Mahape. Okay, so this is the ultimate. Now you may say that, oh, is it only CMD? Absolutely not. CMD did help from time to time to to actually fine tune our hypothesis, but it's our our brain, or rather the mind of a chemist, which actually led to a molecule like this. So I'm not just talking in the air. We do have a proof, and then there are many other molecules which we eventually will develop. And we just want to make sure that we do what is right, as in not just the academic research, but do something which will make there will be some cure for some diseases which we would want to see that happening, right? So despite the limitations and pitfalls, CMD has emerged as a trustworthy discipline for sure. There are several success stories, and I have shared many of them with you. The newer methods, better input data, accurate biological measurements, and then computer, very super computational power, MLDL approaches, and other approaches, they have contributed to the enhanced accuracy of predictions, which is what we would want. And then understanding the limitations of these techniques is crucial, without which it will be just an hype and not just the hope, right? So this is with this. Thank you so much for patient listening. If you have any questions, please feel free to ask. I can show you some of these videos as well, very interesting ones, if you allow me. It's 1 hour and few more minutes. So do you want to go with the videos first and then the question answer, or what? Either way is fine with me.
We can do it otherwise. We can first proceed with question answer session,
Okay.
and then we can go for the videos if you don't mind. Is it fine?
Yeah. Videos, I mean, they are like few minutes videos, like 2 minutes,
Yeah.
3 minutes. They're not big. They're not big.
Okay. So thank you so much, sir, for your enlightening talk on computer-aided drug design, or as you rightly pointed out, it is a computer-aided molecular design. We will now move on to the question answer session. I request Dr. Harish Kundekar, sir, to proceed with the same. Thank you.
Ma'am, and thank you, Prashant sir, for this very informative talk. It was indeed a wonderful presentation, and despite your busy schedule and your personal and professional commitments, you have come up with such a wonderful talk. Thank you so much for it, sir.
Always a pleasure, Harish.
Okay, I would now like to take a few questions from the participants in this Q&A session. Okay, so the first question is from our own faculty, Dr. Nutan Rao, and she's asking how reliable are the free softwares that are available for CAMD as compared to the licensed ones?
Okay, as in, see, when it comes to licensed software, I mean, those are proprietary technologies. So they would have done, because they want to sell the software, and the price of the software is awfully high, although they give 95% discount to the academic institutions. So we can always go, but that price itself is still high, right? So in that case, obviously, I mean, I would say without without even thinking for a moment, that the proprietary softwares will definitely do a wonderful job. But then when it comes to this free softwares, and many a times these free softwares, say something like Autodoc is very well respected, although it's little tricky to use, but once you learn the docking poses which you get and the binding energy predictions, they're pretty reliable, believe me. And then there are many other, like I told you, GitHub, there are many programs available, and these are open source codes. So people who are so passionate about what they are doing, so they keep on improving these. So to start with, these free softwares, because they are designed for general, average people who cannot buy, or average researchers who cannot buy, or who do not have these kind of money. So they still do well. Like I said, it's just that you have to find out the right one for your own need or suit. I mean, you need to talk to people who have used some of these tools, and you will get the good guidance. There are tools for molecular dynamics. There are many molecular dynamics software programs. Although some of these are Linux based, but once you know how to use them, believe me, it's just they're very reliable. I mean, I personally use many of them regularly. So not that we have everything, we have some of them, but the proprietary softwares. But if you use the right tool, your predictions can be near accurate. And thanks to the scientific community who makes sure that these tools are made available to students for understanding, for learning, and there is no harm in using this. But wherever you are using these tools, you should cite those papers. That's the only thing they would want. In fact, my own some of my own papers now target fishing. It's just a really complicated task. So I use Pharm Mapper. I have used some others as well, but Pharm Mapper is pretty reliable. There is another thing called Tarface Doc, which I used to earlier. Even Swiss Admi has one of these target fishing tools. So you can try 2 or 3, and you can you can either include all those results in your thesis, in your paper, or just stick to one. Farm Mapper is good. So you need to talk to people which one to use, because there are many. Yes.
Okay, thank you, sir. The next question is from again from our faculty of Oriental College of Pharmacy, Dr. Amjad Ali. He wants to ask, can you tell us a bit about a licensed software that is available for CAD, which is versatile and at the same time economically convenient?
Perfect. In fact, now if you look at Schrodinger is a big name for sure. I have used Schrodinger all these years, but I Schrodinger is way too expensive, and don't quote me anywhere. Nothing against Schrodinger, but Raghu, if you are listening to this, I'm so sorry when I say this, but your softwares are expensive. I would recommend molecular operating environment, which is now if you look at Schrodinger is like in bits and pieces, so modules will have to purchase. But molecular operating environment comes as one full package. And in fact, at ICT, we have initiated process to buy molecular operating environment for our own purpose. We have bought it for all 3 campuses. It's not very expensive. Probably 1 year's license will cost you somewhere between 1.8 to 2 lakhs, which is not bad. If you have properly, planned, but you have everything, you get everything. It's not module based. Of course, it is token based. And if an academic institution buys that, so if you buy 3 tokens, they'll give you 3 tokens free, because for teaching for free. So molecular operating environment is what I would recommend for sure. I've been using molecular operating environment since 2002. The first time when Raghu started working with Tata Elxsi, he was the one who came to ICT and sold us. In fact, my own part of my PhD work was done using molecular operating environment, where I did comparative protein modeling of Mycobacterium avium complex dihydrofolate reductase. So yeah, molecular operating environment is the one. There is another one coming up from, there is this, I can refer, I don't exactly recall the name, but somebody contacted me. In fact, 2019, we had a seminar from them as well. So they have also come up with a software where they have combined everything, and it's pretty reasonable. I can share that information with Harish, and you can take it.
Okay, thank you, sir.
Yes.
The next question is from Mr. Umair Ahmed, and he's asking, can you name a few government organizations where we can perform cell line activity of synthesized compounds?
Meaning anti-cancer, you mean anti-cancer, right? To start with, that's probably the.
Yeah, possibly he has not mentioned it. He just
Yeah,
mentioned
but
about cell line activity.
I would assume it's anti-cancer. Of course, you can do anti-diabetic, anti-obesity as well in cell lines. So one is ICT for sure. Then Actrec is another place, but then I think they do. It does cost a lot of money. In fact, in my group we are setting up MTT assays, and then probably maybe few months down the line we'll be able to do something like this at nominal charge, which will only cost you for consumables, right? Because for everything you require consumables. So I'm just trying to, because I have struggled myself a lot. So that's where I would want this to make available to those who are looking for that. We have done that previously in my previous organization. Very nominal cost, maybe 100 rupees or 150 rupees per compound. We have done that. We have done that. Again, this is not for business or anything. It's just for the consumables. But then at the same time, you'll also have to put our name in the because it's just yeah. So, anyways, we can discuss about that. But we are going to do that for sure. Then you can also send your compounds to CDRI. That's for sure. CDRI Lucknow. They do high content screening. So if you contact right people, they will be able to do this for sure. ICER is also there, but then you will have to contact individual ICER. I would say ICER in Pune. They do that. That's probably the best of all the ICERs. Probably most developed, best in this is most developed because it started quite early. So you can talk to individual PIs there, and people are ready. You just have to ask. Believe me, you just have to ask. Earlier, NCL used to have anti-tubercular activities, studies, but nowadays they I think there was some funding issue. They did not. In fact, majority of my studies, I had collaborators from for anti-tubercular activity. I have collaborators from South Africa, Vinayak Singh. So he does it for free. Then earlier there used to be one program in Australia for anti-tubercular, not antibacterial and antifungal, OSDD program. No, OSDD was in India. There was another one. I forgot the name. That was long ago. So they also used to do it for free. So there are a lot of places depending on what you would want to do. So if you write to me, I can suggest in anything in particular. I can suggest anti-cancer. Yes, we can do ourselves. Not immediately, maybe 2 months down the line.
Okay, thank you so much, sir, and thank you for your initiative for the anti-cancer testing, for making it easily available to
Yes.
people who really want to work in the area of drug design for anti-cancer molecules.
Yes.
And then there is a series of questions from Mrs. Sonal Parmar and Pritesh Patel. So I'll just compile all of them together and ask as
Sure.
one question to you.
Mm-hmm.
Can you recommend some free energy minimization softwares and some softwares for the BBB? And then they have also asked about softwares for toxicity prediction. So I think that was a question that was already answered by Sir in his talk. And whether Maven Playground HERG toxicity prediction is a good software to be trusted. So basically, they have
See
asked
now,
about direct questions about the software.
yes. First, I'll talk about the BBB permeation software. You can just Google it, BBB permeation web tool or something. It will take you to some publications, and there you will get the link. And many of those they work, and the output is binary, either plus BBB minus BBB. It will not give you some certain percentage or not. So either yes or no. It will just tell you that. And in fact, BBB permeation at times becomes very easy in case you just have, if you calculate the TPSA or topological polar surface area, if that surface area is above 80 angstrom square, then the molecule is not likely to cross blood-brain barrier. It will be orally bioavailable if it is less than between 80 and 140. If it is less than 80, then your molecule is likely to be BBB permeable. If it is less than 60, definitely yes. But if your molecule has got an acidic functionality, it will probably not cross blood-brain barrier because acids are prevented from entering in the brain unless there is some active transport. But bases, they just pass like that. That's one. Regarding free energy minimization, so many of the softwares which are available for molecular dynamics, you can actually search on their website. There is one called VMD. Then there is another one for Nomads or something. So there are many others, which, like I said, they are Linux-based systems, But if you go to their website, so they will have a separate module for free energy studies for sure, right? So there is another one, free energy perturbation. So you can the best version you can get in is Schrodinger for free energy perturbation methods. I haven't used them myself because, of course, for the computational power, but someday I would want to use it and try it myself. But like I said, there are some, and if you again search in GitHub, you might get some code which will be an addendum to the already available softwares, which essentially can help you in doing those tasks. Even in MOU, if you purchase a licensed version of MOU, people write a lot of SVL script. SVL is their language. So those scripts can be downloaded from their website, and only licensed users can download those, and you can use them for doing some customized tasks. Other one would be, I would recommend NIME. If you have used NIME, it's a free chem informatics tool. NIME for Constance Information Miner. It was actually funded by my company, the company which I used to work for in Constance in Germany, that is Nycomed Pharma. It's actually, this is like, you have to develop the workflow. And many of the Schrodinger workflows are based on NIME nodes. So a node is a program. So depending on whether you want to minimize the structures and then dock and then screen for physicochemical activity, so you just have to connect those nodes, give the input node, and then all the functions are executed by those small programs. So you select a node, meaning you are selecting a program, which when receives an input gives you the desired output, the node for which it is designed for. So use NIME, believe me, you don't have to look here and there. It has many tools for other chem informatics based studies as well. Explore NIME. There are a lot of videos. Just type KNIME and it will take you to the website. You can download NIME. Then you can also yourself watch those videos, go through their course. It's all free. It's all free. And they have user workshops. You can post in your particular requirement. Somebody may write code for you and make that node available for you. People do that all the time. You just have to make people know that you are looking for something. And for students, faculty, people will help.
Okay, thank you so much for that answer. And one last question that I would like to ask you is, you talked about bioinformatics, Chem informatics, etc. So nowadays, bioinformatics is a booming field, and more bioinformaticians are involved in utilizing softwares related to CMD. So how would you differentiate between the roles of a bioinformatician and a medicinal chemist?
Well, I mean, these are two different skill sets. When you say bioinformatics, you need to have familiarity with specialized tools like Perl, and then there are various algorithms which are specifically designed for mining the say bioinformatics based or biological information. So it's like DNA microarray data, meaning when you have a particular type of cell is treated with a molecule. So then what are the genes which are overexpressed and what are the genes which are underexpressed and the set of genes which do not get affected. So all that, it's a huge information data. And when you talk about bioinformatics, the pathway analysis is another interesting feature where people, in fact, if you look at drug repurposing using genomic based approaches, there are more success stories than the ones from the chemical based approaches, because at times it is very straightforward. You get this data from those instruments, and that data is really used. So all you need is you need to write a script to look for particular individual information. I mean, not necessary that you should have coding knowledge, or so you can still do without coding knowledge. But if you have coding knowledge, things become very easy. So if you talk, I mean, if you are a medicinal chemist and if you also learn the bioinformatics tools, believe me, you will be, you will be able to do wonders actually. I mean, we shy away from things which we think are complicated. I mean, I personally haven't used bioinformatics much. The hardcore bioinformatics, meaning what you do, say using when I build a comparative protein model or a homology model, it's actually bioinformatics, where I'm aligning protein sequences, trying to see how much similar they are, how much, what are the protein folds and everything. So that comes under the purview of bioinformatics. But then I use it for medicinal chemistry purpose, where I use docking and then see how my molecules are binding to the theoretical model. So yeah, if you if you really learn, there are particular courses. There is a course from JNU, and then even the Bioinformatics Institute of India in Pune, in the NCL campus. So they have specialized courses. And then, in fact, in Gujarat, I think I've seen some course which DBT offers. And that course, if you are enrolled, if you are selected for that course, you also get a fellowship because they want to promote mathematics in students. Meaning, students should get trained because this is what we need at this point of time. I mean, this century is going to be the century of the biologics. Earlier century was century of small molecule drugs. So if you are associated into bioinformatics, anything related to biologics, which we did not really talk much about today. So believe me, that is the future.
Okay, thank you so much, sir. Just allow me to take one last question, and it comes from our faculty member, Dr. Muhib Khan. He's asking in Mumbai, from where can we do docking study at reasonable cost? This is from Dr. Muhib Khan.
Okay. We can help for sure. As in earlier, Dr. Coutinho's group, they used to help anybody and everybody who approached them. Now, Elvis, sir, student is he's working as a faculty there. So approach Elvis, and Elvis will help you. You can come to us as well. We can help in any possible way. Now I have got my students. My group is also increasing. In fact, if you approach me after a month, I'll have 1 or 2 students who are into this field. Meaning, I do it all the time, but then at times I'm too busy to do it. But then my students will help you. So you can approach us. You can approach to even like I said, Bombay College of Pharmacy. Elvis is there. He is molecular dynamics expert. So anything related to molecular dynamics, they do have a cluster of computers which can if you guys agree to work together, Elvis can definitely help. I haven't really spoken to him, but he wouldn't mind me recommending him for these studies, and he will be happy to help.
Thank you so much, sir, for all the answers. And I now request Dr. Nutan Rao to take over.
Thank you so much, Harishka, for conducting the session. Once again, thank you so much, Dr. Prashant Thakur, sir, for your insights with respect to the computer-aided molecular design. It was indeed a thought-provoking session and will definitely spark interest in young minds with respect to modern computer-aided molecular discovery research. Rather than drug discovery, we can call it as molecular discovery research. So all the attendees here are informed that the link for feedback is posted in the. Kindly fill in the details carefully since the certificate will be processed accordingly. The link for feedback would be open till 4 p.m. only, after which it will be closed for accepting the responses. So, sir, you were talking about some videos, so we can just go through that.
Just 1 or 2 videos which will
sure.
which will I hope I'll be able to share them. Let me just get back. Okay. I think I'll just share the screen. Hopefully that will work. Are you able to see Swiss admin?
Yes, sir, yes.
Yeah. So now when you say, I'll just quickly demonstrate how to do it. If you go to ChemDraw, are you able to see ChemDraw?
Yes, sir.
Yeah. So what I've done is I have drawn structure of haloperidol, which is an antipsychotic drug. So you select this molecule and then go to edit and then say copy as SMILES. Okay, SMILES is simplified molecular input line entry system, which is the one-dimensional representation. Copy this and then go to Swiss admin and just go to enter a list of SMILES here. Just say Control V, paste, and just say run. It will take less than 10 seconds. And see, now all the activities are predicted for this particular molecule, right? This is probably the easiest one. In addition to that, for target fishing, you need to go to Swiss target prediction. Okay, here also it will give you select a species for either the rat or the mice or the Homo sapiens. So either you draw the structure or again do the same thing, that is paste the smiles, and then it will appear, the target will, meaning the structure will appear, and you just say predict targets. So after a while, it will give you the list of potential targets. Now it's already a known thing. Its target is dopamine D2 receptor. So you will see that it's a known target. It takes little time, but it will happen. So this was one. Maybe you can try it yourself. I'll go to the, yeah, it will ask you say top 15 targets. Yeah, it's a, now you see they're all GPCRs. Dopamine D2 receptor is a GPCR. So you probably will see them in here, dopamine D1 receptor, serotonin 2B receptor, dopamine D5 receptor, alpha 2 adrenergic receptor. They're very close, right? So you will see the probability is 100%. So, I mean, this is a known target, so I wouldn't really, kind of, I'm not surprised, but I'll take you to Protox as well. Let me just show you. Protox 2. Maybe I'll just, all you have to do is just go to Google and say go to Protox 2. Here also go to Tox prediction, and then just paste the smiles. So you need smiles for anything and everything, and then the structure should appear. Just say click on smiles. The structure will change to what you have pasted. Then you can select from your hepatotoxicity, carcinogenicity, immunotoxicity, mutagenicity. If you don't select anything, it will give you all of it. All. It takes time, but it will give you the probability of that particular toxicity endpoint. This is one very interesting thing. Then I'll go to one video which is about the molecular dynamics. So what molecular dynamics is, probably you know already, but I'll still take you. Don't worry if you do not hear. It's just a music, I guess. I'll just put it on. Don't worry about this background thing. I'll probably just. Yeah, so here the point is with the help of molecular dynamics, you can predict the movement of atoms in future, because if given the XYZ coordinates and the connectivity and the force of each atom, it uses Newton's laws of motion to predict the future position after a certain time, right? So here what we do is we try to capture the molecular motion, and if you look at molecular dynamics, meaning the smallest or rather the simplest bond is CH bond when you talk about organic molecules or even biomolecules. So CH bond takes around 10 raised to minus 14 seconds to vibrate. So what we do is we take the time step, which is 10 times the smaller than the time taken by CH bond to vibrate. So we take 1 femtosecond as the time step. So any and every motion of each of these vibrations is captured, and this is what molecular dynamics is all about, to predict the like how this molecule will evolve. Each of these atom of this macromolecule will evolve. So that includes. So here you see the stapled peptide, its helical conformation is stable, whereas the wild type is free to move, right? So this is the beauty of molecular dynamics. Maybe I would want you to see the last video, probably 2 or 3 minutes video, which talks about the I would say the cryo-electron microscopy. It's 2-3 minutes video.
All
Is
right.
that okay? Shall I play it?
Yes, 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 sir.
Yeah. Okay. You should see this. This is amazing. No, this one is Schrodinger. I don't want to show this. I'll take you to the next one. Yeah, this is the one. This is 5 minutes, but it's worth it. I'll stop it in between. Don't worry. Or just hold on. I'll probably show you the next one. This is 2 minutes 36 seconds. No, this is really cool. If I have to tell you that, because it gives you the dynamic motion or the dynamic pictures of molecules, macromolecules in different orientations, and 3 scientists who contributed differently to this complete technique, they got Nobel Prize for this discovery. So yeah, a lot of things are happening, and that this is how this computer-aided molecular design field will progress, and it has already entered in almost all the areas of science, and that's where one must be aware of these techniques and use to the fullest wherever possible. That's all from my side.
It was wonderful journey, sir. Right from your talk, from the videos, whatever you have shown, also the personal experiences, as well as the publications. JMC 4 publications till now. It's like just amazing. It's an amazing achievement. So congratulations to you, sir. And we are really blessed to have a speaker, resource person like you. And in future, definitely, we would like to hear you once again. Uh, Maybe we can have certain workshops related to that.
Yes.
So that
Anytime,
would be
anytime.
useful for the students as well as the faculty members in their research.
Yes, yes.
So just a quick question.
Yes.
Can you compare the X-ray crystallography with the cryo-electron this thing? So what is the exact difference? Because in X-ray crystallography, you come to know the coordinates, right?
Yes.
All the coordinates, X, Y,
Yes.
Z coordinates. So what is the difference?
So here also you get the coordinates. The only interesting part is now you see when you have the molecule in the 2D crystal, so some molecules will be oriented in one particular fashion, some molecules will be oriented in different fashions, and thousands of these molecules. So the main work is done by the software, the computer algorithm to come up with the 3D structure. But in my personal experience, of course, I haven't used any of the cryo-electron microscopy structure, but their resolution is a bit less. That is one problem. But like I said, this is just a technique which started like few years ago. Although the guys work for more than 25 years on this technique, but the commonplace use has started very recently. So then there will be improvements further. So the resolution is slightly less than what you otherwise see in the X-ray structures. But if something like some very big protein which couldn't really be crystallized, now we have the structure available from cryo-electron microscopy, and we can always work on that and take it forward. So we do have some starting point to start with. So
since the word cryo is used, there is something to do with freezing. So how does it really help to have the structure known?
So what they do is they make a thin film, and then they use liquid nitrogen to freeze that film. So the molecular motion is frozen in that liquid nitrogen. So the molecules are captured, and this is like taking a picture. So when this electron beam passes through that, just the way we take CM or TM structures, this is actually a TM structure, if I have to tell you that way. So then you get the picture, and that picture is processed by the computer algorithm based on some prior knowledge on the amino acid sequence and the other things. And so they put everything together, and finally we get the cryo-electron microscopy structure. In fact, in PDB, there are a lot of structures which have originated from cryo-electron microscopy. But these structures are awfully big because that's what cryo-electron microscopy is more useful for, because whatever is possible with X-ray structure, people would do that. And now we have high throughput X-ray crystallography. So things have changed. And if you see in last last 3 years or so, the number of crystal structures it actually went beyond 100,000. It is very close to touching 200,000. So the speed has increased tremendously.
It is very interesting actually.
Yes.
And something like is called as proof for thought. So it is there, cryo-electron microscope.
Yes.
Wonderful.
I mean, at times I believe we limit ourselves by our own thoughts. We have to just come out and explore things are possible. Talk to people. If you're really passionate about something, talk to people. People will help you. I have experienced that all the time. Not that I know everything, or not that all my publications are only my group's efforts. Absolutely not. I credit everything to my collaborators, very kind collaborators. I've been very fortunate to get collaborators. And that's exactly what I would want to replicate. Whomsoever approaches us, if we are in a position to help someone, we would do that.
So collaboration is the key.
Yes, that is the key. And many people fail to understand. And you cannot do everything all by yourself, given the funding situation, given this current pandemic. So everyone, not that, I mean, many times we think that why would foreign collaborator would agree to what I have to do or absolutely not. In fact, we have tried and tested. And every time we approach somebody, we have got the things done. In fact, Dr. Toraskar from Bharati Vidyapit, in 2014-15, we approached one guy, Dr. Claudio Sukuran, who is probably one of the best guys for carbonic anhydrase field. So we approached, she had molecules which had good anti-cancer activity, and she believed that there was some carbonic anhydrase 9 connection. So we just wrote to him, he said, send your compounds. We sent the compounds, two different series, and we got two different papers. One is in ACS chemical Medchem letters, and the other one was in Journal of Enzyme Inhibition and Medicinal Chemistry. So yeah, people help. We just have to approach.
Yeah, connecting is important.
Yes.
And people like you help a lot. So that is going to be useful.
Because a lot of people help me, so I just want to do the same thing. Because at least while I'm in ICT, I'm in a condition to help people whomsoever approaches in whatsoever possible way. I would definitely look forward to that and do it.
Thank you so much, sir. So we are waiting for this thing also, your talks also, maybe some workshops, as well as
Anytime,
collaboration.
anytime, ma'am. Anytime, not a problem. Anytime.
Thank you, sir.
Thank you
So
so much.
now we are at the end of the session. Now I would like to request Dr. Amjad Ali, Associate Professor in Pharmaceutical Chemistry, to present the vote of thanks.
Wow, good to see you, sir.
Thank you, sir. Thank
You
you.
was, I think you are from first batch of Bharati Vidyapit master students, right?
Yes, sir. Yes, sir.
Yes, I remember. I just completed my PhD. I taught them some.
Sir, you have taught us
subject.
CAD over there.
Yes. I remember in 2003. Long time actually, almost 20 years. Good to see you, sir. Very nice. I'm very happy.
Thank you, sir. Thank you. Thank you, Nutan madam. Dignitaries present for today's seminar: Professor Prashant Khatkar sir, speaker for the webinar. Professor, Department of Pharmaceutical Sciences and Technology, ICT, Mumbai. Dr. Sudha Rathore madam, principal and convener of the webinar. Dr. Mrs. Nutan Rao, HOD Pharm Chem and coordinator of the webinar. All HODs, faculty members, delegates from industry and academia, and my dear students. Good afternoon to all. It is my pleasure to for this memorable webinar. All praises and thanks are to Almighty for showering His blessings for everything and to organize successfully today's webinar. It is my duty to thank our patron, Honorable Mrs. Humaira Javed Khan Madam, President OES, Oriental Education Society, for permission to organize this wonderful webinar. I thank our patron, Honorable Dr. Azim Javed Khan Sir, Treasurer OES, and Honorable Mr. Wasim Khan Sir, General Secretary OES, who has given financial assistance wholeheartedly. I am indebted to Honorable Dr. Haidare Karyo OES inspiration to conduct the webinar. I express gratitude for our dynamic principal, Dr. Mrs. Sudha Rathore, convener of the program, for her kind presence, guidance, and constant cooperation for the success of webinar. On behalf of our management, principal madam, staff members, and students, my sincere thanks are to the speaker of the webinar, Professor. Sir, you have explained CAD in a very way with lot of examples, graphics. So it was wonderful, sir. Thanks a lot. Sir, 2 lines for you. जहां भी जाएगा वो रोशनी लुटाएगा, जहां भी जाएगा वो रोशनी लुटाएगा, किसी चिराग का अपना मका नहीं होता. So thank you, sir, for accepting the invitation, sparing your valuable time to make this webinar a grand success. I am thankful to Dr. Mrs. Nutan Rao, Associate Professor in QA and HOD Pharm Chem, for coordinating the webinar. I thank to members of the organizing committee, Dr. Harish Kundekar, sir, Ansari sir, Mr. Suraj Gupta sir, Dr. Mansi Golkar madam, Ms. Swarnali Manik madam. Without these committee members' dedication and enthusiastic work, it was not possible to complete the work. I am thankful to all teaching staff members for their presence, kind help and support. The last but not the least, I thank 2 delegates from industry and academia. all the students of M.Pharm and B.Pharm of all pharmacy colleges for their presence, enthusiasm, and maintaining the discipline throughout the webinar. I'm thankful to IT person, Mr. Rahil, for his support. Once again, thanks to one and all. Jai Maharashtra, Jai Hind. Thank you.
Thank you so much. Thank you.
Over to you, Nutan madam.
Yes, sir. Thank you so much. So we all can leave.